that bile exits the gallbladder through what's called the cystic duct, which is on right here. Cystic duct. The cystic duct is going to join the common hepatic duct. It's going to do this. When the two join, the new duct that's formed is called the common bile duct. The common bile duct is going to pass down through the head of the pancreas like this. And then within the head of the pancreas, it is going to be joined by the pancreatic duct. Pancreatic duct is a duct that drains exocrine secretions from the pancreas. Okay. You have all these little tributaries like this, draining into the pancreatic duct. The pancreatic duct is going to join the common bile duct.
Are you saying bile or bowel?
Bile, B-I-L-E.
Okay, that's what I thought.
Now, when the common bile duct joins the pancreatic duct, they form a larger duct. It's called the ampulla of Vata. An ampulla is considered a dilated region of a tubular structure. So the ampulla of Vata, V-A-T-E-R, it's like water except a V instead of a W. That ampulla of Vata is going to then carry bile from the common bile duct and pancreatic secretions together into the duodenum. It will empty into the duodenum. Now, at the point at which it enters the duodenal lumen, there's a little nipple-like projection of mucosa, as I've drawn here. That nipple-like projection is called the major papilla. The reason we have that papilla is that deep to the surface, there is a ring of smooth muscle which forms a sphincter around the opening. That's called the sphincter of Oddi. So the sphincter of Oddi is going to control the flow of these secretions from the ampulla of Vata. What do you want to call it?
There is also called the hepatopancreatic ampulla.
If you can spell it correctly, you can use that term.
Okay.
If you can, most people don't use that term because it's long and messy.
Right.
So this is an example where one of these eponyms is preferred.
Right.
All right. Now, most of the time, this sphincter of Oddi is contracted, so that when bile begins to come down from the liver toward the duodenum, it backs up. It's going to back up into the gallbladder, where the bile is stored and concentrated. Okay? So as long as that sphincter of Oddi is closed, as long as that smooth muscle is contracted, then the bile that's being produced is going to back up into the gallbladder where it's simply stored. Now, the reason we have a major duodenal papilla right here, and we call it major, is that there's also a minor duodenal papilla. Because just like there's we call this the pancreatic duct, there's an accessory pancreatic duct in many cases that will open separately into the duodenum, and that accessory pancreatic duct can be associated with a minor duodenal papilla. But we're not going to worry about the accessory duct. We're going to worry about the major pancreatic duct. All right. If we look at the mucosal surface of not only the duodenum, but the jejunum and ileum, the entire small intestine, what you're going to see is that the mucosa is thrown into folds. Now, they're not longitudinal folds like we see in the stomach. They're more circular folds that kind of look like speed bumps that slows down the movement of chyme as it passes along the small intestine. Okay? So you see the difference in the arrangement of these folds compared to the stomach? They're not longitudinal, they're circularly arranged. Now I use the term chyme, C-H-Y-M-E. Last lecture, we said that when food was mixed with saliva, it formed a bolus. That bolus is going to be passed down into the stomach, and when that bolus mixes with the gastric juice, it becomes chyme, C-H-Y-M-E. And so it's chyme that's going to pass from the stomach into the small intestine. Okay. I mentioned that these little speed bumps, okay, these folds within the mucosa, kind of slow down the passage of the chyme. These folds within the small intestine have a name. They're called plicae, that's P-L-I-C-A-E, circularis. Plicae circularis. Now, these folds, these plicae circularis, include not only mucosa, but submucosa. Okay? They don't include the muscularis externa or the serosa, but they do include the mucosa and submucosa. And part of the purpose of these folds is not only to slow down the movement of chyme, but to increase surface area. You see, the small intestine, and particularly the duodenum, is the major site for digestion and absorption. This is the site where nutrients are broken down into building blocks, and these building blocks are absorbed. So by increasing the surface area to which those building blocks are exposed, you increase the efficiency of absorption. Okay? And in fact, the plicae circularis are only one of 3 adaptations that provide for an increased surface area for absorption. If you look at these plicae circularis, okay, coming off of them are these finger-like extensions of mucosa. These finger-like extensions of mucosa that we see right here are called villi. Okay, so you have plicae circularis, and then you have villi. Singular would be a villus. A villus is a finger-like extension of mucosa that serves to increase surface area for absorption. Notice that the surface of the villi are lined by a simple columnar epithelium. Now that's not surprising because we said that within the GI tract, from the stomach to the rectum, the epithelium is simple columnar. Okay? So if you look at the villi, you have a surface epithelium, and deep to it, the lamina propria. That lamina propria contains capillaries, small arteries and veins, arterioles and venules, as well as lymphatic capillaries. Now, in this location, within the small intestine, the lymphatic capillaries have a special name. We call them lacteals. Lacteal, lacteals. What we're going to see is that the vascular capillaries are going to serve the purpose of absorption of sugars and amino acids, whereas the lacteals are going to provide for absorption of fat. We'll come to that later. Now, if we take a little portion of the epithelium like we have here, and then magnify it to a higher power, you'll see that the individual cells, the absorptive cells that line the surface of these villi, have an apical surface modification, microvilli. Again, the purpose of these microvilli is simply to increase surface area for absorption of those nutrients. Okay? So we said plicae circularis, villi, microvilli. Okay. These are the three surface modifications that serve to increase surface area for absorption. That's the main role of the small intestine, digestion and absorption. So when nutrients are absorbed across the surface of these microvilli, they're going to pass through the apical surface as well as the basolateral membrane of those cells into the lamina propria, where they'll get absorbed either through the vascular capillaries and through lacteals. Now, if the nutrient is absorbed into the vascular system, if we're talking about the small intestine, those nutrients are going to travel from capillaries ultimately to the superior mesenteric vein. Remember, the superior mesenteric vein is the vein that drains the small intestine. The superior mesenteric vein is going to carry those nutrients into the portal vein, and from the portal vein into the liver. So those nutrients are going to first pass through the liver before reaching the systemic circulation. So they're showing here a superior mesenteric vein carrying blood to the hepatic portal vessel. All right. If we look at the duodenum, underneath the microscope, we're going to see several different features. In fact, we're going to see several different types of cells. The major cell type that lines the surface of these villi is the simple absorptive cell. Its entire role is simply absorption of nutrients. But scattered between those absorptive cells, you're going to have goblet cells. In fact, that's one of the features of the small intestine, is you have absorptive cells with intermittent goblet cells. When you first look at a slide, a field of view, and you see that the surface epithelium is simple columnar, you know that this isn't the mouth, it's not the esophagus, this has to be somewhere between the stomach and the rectum. When you see the mucosal surface thrown into these villi, these finger-like projections, now you're saying this is small intestine. So in the small intestine, you see these villi that extend up and down like this. And in some regions, they're very close, they're very crowded. Okay? Extending up to the surface between the villi are intestinal glands. So what we're going to see is that intestinal glands are going to invaginate down into the lamina propria. Like this. and they're going to open onto the surface between villi. Okay. Now, in the small intestine, and some people do this in the large intestine too, we'll call these intestinal glands crypts of Lieberkühn. Crypts of Lieberkühn. This is a German word, Lieberkuhn, and to spell it correctly, you have to put this thing with 2 dots on top of the U. Crypts of Lieberkühn.
So we're going to utilize those.
What's that?
So can we
Yes.
just call them intestinal glands, or we have to call them?
No, in the small intestine, call them crypts of Lieberkühn.
Okay.
Now, in the large intestine, more often they're simply called intestinal glands. But in the small intestine, they're more often referred to as crypts of Lieberkühn. And these crypts of Lieberkühn produce an alkaline mucus that helps to neutralize the acidic pH of the chyme. Remember, the chyme is coming from the stomach, where the acid environment is more acidic than anywhere else in the body. That the gastric juice has a pH of around 1 to 2. The problem is that when that chyme reaches the small intestine, the digestive enzymes present within the lumen function best at a higher pH. So that material has to be neutralized. So one of the purposes of this alkaline mucus is to neutralize the acidic pH of the chyme. So we see the intestinal glands extending down into the lamina propria. All right. These glands have scattered enteroendocrine cells. Remember from last class that enteroendocrine cells are cells that are specialized to produce hormones. Either hormones that act locally, as was the case with gastrin, or traditional hormones that then enter into the bloodstream. One type of enteroendocrine cell